Good morning, everyone. How are all of you? Audible, visible, first pin your response and also write up to where it is completed. Very good morning. ER completed, very good. So we have to say about Golgi body, vacuole, lysosome, right? And mitochondria. Let's put a target for today for these 4 cell organelles. Let's see up to where it is going to finish, right? So let's start with Golgi complex. So what is Golgi complex or Golgi bodies? What is Golgi complex or Golgi body, right? So it is densely stand a reticular structure reticular structure present near the nucleus. Right, present near the nucleus. Getting the idea or not? Now, first observed by Camillo Golgi, C. Golgi. And on honor of his name, we called it which one? Golgi body or Golgi complex. It is also called lipochondria and dictyosome. This dictyosome word we use in which one? Plant Golgi complex. So in plant Golgi complex is called dictyosome. Right? So now I am going to write about the component of Golgi complex. As I told, same component present in ER and Golgi complex, that is what? First is cisternae. Second one is what? Vesicle. And third one is which one? Tubules. But in case of Golgi complex, these three structures are assembled together and making a complex. Right? Now NCERT wrote about cisternae as well. So what they wrote about the cisternae, I am writing here, they are writing it is flat tubular structure, stacked over each other. And number of cisternae is variable. Number of cisternae is variable from Golgi body to Golgi body. So I am drawing the diagram. You just look here. Suppose that this is a cell and here is nucleus. Now I am drawing the cisternae of the Golgi complex, major component. So you just look, it is like this. So it is a which one? Discoid structure. Na? If we talk about cistern, is what basically? It is discoid, disc-like a structure. Got the idea? Na? It is a disc-like a structure. Disc-like a structure. And it is assembled like this, in a stack, one above the other. And the number of this cisternae is variable. Getting the things? And these stack like a structure is interconnected also. These stack like a structure, one, second, third, and I'm just drawing one more. These are a stack like a structure, interconnected also. So this is a Golgi complex. Now NCERT also wrote the diameter of cisternae. Tell me, what is the diameter NCERT wrote? Pin the answer. What diameter of cisternae written in your textbook? First, try to answer without help of NCERT. If you're not able to answer, then take the help of NCERT. So what is 0.521 watt? What is the unit? Unit also write. Micrometer. Very good. Until unless you will not write every complete information, problem will arise, na? Micrometer. Got the idea? So the diameter of the cistern is which one? 0.1521 micrometer. Now look. This cisternae is discoid in shape. So toward the nucleus, it is nucleus. It is nucleus. So toward the nucleus, it is convex. And toward the plasma membrane, it is concave. Are you agree or not? You see the structure. Toward the plasma membrane, this is what? Plasma membrane. So toward the nucleus it is convex and toward the plasma membrane it is concave. So this convex area is also called cis face. Cis face, or it is also called forming face. Forming face. Got the idea? And concave area is also called which one? Transface. Transface or maturing face. Transface or maturing face. Are you getting the idea? So concave face is called cis face or forming face. Concave face is called transface or maturing face. Got the idea? So can we say Golgi body is a cell organelle with polarity? Cis and trans is there. Is a cell organelle with polarity. Polarity we are telling due to two phase, cis phase and trans phase. So you got about the structure of the Golgi body. It is made up of which one? Flattened discoid structure, and they are stack over each other. Is it clear in the mind? That is stack over each other and interconnected with each other also. Got the idea? So now we are going to talk about function of Golgi complex. A structure we have seen. Now we are going to talk about function of Golgi complex. So what to write about the function? Three major functions I am writing. First, packaging. Second function I am writing, cell secretion. Third function I am writing, chemical modification. That is the function written in NCERT. And chemical modification, that is what? That is formation of glycoprotein and glycolipid. So attaching carbohydrate with protein and it become glycoprotein, carbohydrate with lipid, it become glycolipid. So these are the function mentioned in NCERT regarding Golgi complex. Beside of that, I am writing some more function. which is not mentioned directly. So I am writing formation of lysosome. Formation of lysosome is also a function of Golgi complex. Another function I am writing, cell plate formation. Cell plate formation is also a which one? Function of which one? Golgi complex. Getting the idea or not? So these are some of the function of Golgi complex I have written here. Now I am just going to deal how it do the chemical modification. How it do the chemical modification. So I am using the diagram, same diagram to make you explain here. Just try to understand. Then you will understand well about the NCERT also. Have a look here. Just give your attention. This is a cell, this is nucleus, this is Golgi body, and here I am drawing endoplasmic reticulum. So you just assume it is which one? ER. It is which one? Endoplasmic reticulum. Now what happened? How they do the chemical modification? Let me tell you, I am explaining it. So what happened? On the surface of which one? Endoplasmic reticulum toward nucleus, ribosome is present. And these ribosome present on the surface of ER perform protein. So what is this? Protein synthesis occur. Protein synthesis occur. So protein synthesis occur on ribosome present on which one? ER. Now what happened? These protein came inside of lumen of ER. These protein came inside the lumen of ER and it is going for packaging. Not packaging, folding. It is going for folding. So folding of protein occur in which one? ER. And folding convert primary structure into secondary structure into tertiary structure. Able to understand or not? So these changes in protein occur in which one? Endoplasmic reticulum. Now these protein get packed in vesicle of ER. So this is a folded protein and this structure is what basically? Vesicle of ER. So who is packed in vesicle of ER? Protein is packed. Protein present outside of ER on ribosome, then enter in ER, then go for folding, then get packed in vesicle of ER. If you want to know more how it packed in vesicle of ER, so look here. It is what? It is the which one? ER. Lumen of ER. Here who is present? Protein is present. Assume. Now what happened? Just see. These lumen going to become like this. And who enter here? These protein enter. Then they detach from here. After detaching, it become which one? This kind of a cycle. And who is present here? This protein is present here. How this protein came in vesicle of ER? You got the idea? This is lumen. How exvagination occur? Protein enter, then it detach from here, and this protein is packed in which one? This vesicle of ER. Now what happened to the vesicle of ER? These vesicle of ER, try to understand, fuse with which phase? Cis phase. These vesicle of ER fuse with cis phase of Golgi complex. And they're releasing the protein into the cis phase. It is cis phase, na? It is forming phase. Now these protein moving from cis phase toward trans phase. This protein is moving from where to where? Cis phase to trans phase. So protein is moving from cis phase to trans phase. So when protein move from cis phase to trans phase, what happened? Chemical modification occur. This is written in NCERT, na? Chemical modification occur. chemical modification occur. That is, who is forming? Carbohydrate joined with protein, oligosaccharide joined with protein, and who is forming? Glycoprotein is forming. If lipid is coming, I told about protein, if lipid is packing, so who is forming? Glycolipid is forming. Are you getting the idea? So how these chemical modification occur? When it is moving from cis phase to trans phase, what happened? Chemical modification occur in the Golgi complex. So entire picture came in your mind or not? Now what happened finally? Finally, these protein packed in a vesicle, chemically modified, packed in a vesicle called Golgi vesicle. Golgi vesicle. So what are the chemical is coming after modification? They are packing in Golgi vesicle, and now Golgi vesicle transport this packed chemically modified protein or lipid either at intracellular target or to extracellular target. So extracellular target is fused with the plasma membrane and material come out. It is called secretion. So Golgi body is the cell organelle who transport the chemically modified protein and lipid either to intra or extracellular target. So Golgi body vesicle transport chemically modified protein to intra and extra cellular target. You got the idea? When it is fusing with membrane, mitochondria, it is intracellular target. This protein came here. When fusing with cell membrane, it is extracellular target. Secretion occur, and these putting the chemically modified protein or lipid into a vesicle is called packaging. So how it is doing packaging, secretion and chemical modification, everything came in the mind or not? And that is all about which cell organelle? Golgi body. I hope you understand it. So coordinated function you have seen na? From ER, outside protein form, then how it came in ER. After coming in ER, it go for folding. After folding, it packed in the vesicle of ER. Vesicle fuse at cis phase. Protein enter in cis phase. When they move towards trans phase, chemical modification occur, glycosylation occur, glycolipid and glycoprotein form. So mainly, which cell organelle is doing the glycosylation? Golgi body. If Golgi body not given in option, you go for which one? I already told earlier, ER. But when, who is not available in option? Golgi body. Clear in the mind? If Golgi body is not given in option, then go for ER. Clear in the mind? So, who is done? Golgi body is over. Now we are going to talk about lysosome. Getting the things or not? Lysosome. So we are starting with the lysosome. Give your attention. What is lysosome? It is membrane bounded vesicle formed by Golgi complex. You just try to understand with this example. These are many vesicle of Golgi complex. Each vesicle have protein or enzyme. These are vesicle of Golgi complex. These are going to form, fuse and forming a structure that is called which one? Lysosome. Are you getting the idea? Forming a structure that is called lysosome. So many vesicles of Golgi body fuse together to form a which one? Lysosome. So what happens? So many, every vesicle have a chemical, na? Right. So what happened? These lysosome lysosome contain more than 50 enzymes, and these enzyme is called acid hydrolase. Acid hydrolase. So more than 50 different enzymes is present in lysosome, and these enzymes are called acid hydrolases. Why we are telling acid hydrolases? Because optimum pH at which, so they work best as, at acidic pH. And that is which pH is the optimum pH? 5 pH. So more than 50 different acid hydrolases present in the which one? Lysosome. Now some example of enzymes. Example. So what NCERT wrote? Hydrolases example, what they wrote? Hydrolases example is, they wrote protease, carbohydrates, lipase. They not wrote word nuclease, but I am writing nuclease. Protease digest protein. Carbohydrates digest carbohydrate. Lipase digest lipid. And nuclease digest nucleic acid. Digest what? Nucleic acid. And many more hydrolytic enzymes are there. Getting the idea? Is it clear? So now a question arises, how lysosome all the time maintain acidic pH? If I ask a question, what the answer will you give? Try to interact. How lysosome maintain acidic pH? Anybody can answer it. Try to answer it. I am just observing your screen. Getting the things? So try to answer. How they maintain acidic pH? No answer came. No answer came. Should I answer? So it maintains the acidic pH. How I am telling? There is a protein present in membrane of lysosome, and that is pump protein. Which protein? Pump protein, and this is cytoplasm. So this pump protein actively pumping H plus ion, proton ion, pump actively. Actively means pumping, actively means expenditure of ATP. Actively H plus ion. And when H plus ion go inside the which one? Lysosome, pH will decrease, it remain acidic, so whenever they find pH is increasing, so they are pumping which one? Proton. And better you know, proton ion concentration if more, pH will what? Basically decrease. So how they maintain the acidic pH? You got the idea. Now I am writing about function of lysosome. So what is the function of lysosome? Intracellular digestion. Because lysosome contain many hydrolytic enzymes, so it is doing what? Basically intracellular digestion. Getting the things? Are you interested to know how it do intracellular digestion? It is not written in NCERT, but if your interest is there, I can say. It will take some more time to stay here, no problem. Right? So, anyone have interest to listen? Should I say NCERT over? Even in NEET question will not come. I am also saying, even in NEET question will not come. But how it do the intracellular digestion? Got the idea? Look. So, I am just writing. It will take another 3-4 minutes, not much. So, I am writing lysosome is a pleomorphic cell organelle. Pleomorphic means it have a different structure in different physiological state. It have a different structure in different physiological state. Look here. Suppose that this is a lysosome. Sorry, this is a cell. Assume this is a amoeba cell. And in amoeba, assume this is the lysosome. In this lysosome, which enzyme is there? Hydrolytic enzyme. It is primary lysosome. So lysosome have 3 different physiological state. One is called primary lysosome. Another is called secondary lysosome. And third one is called residual body. And these secondary lysosome is of 2 types, autophagosome and heterophagosome. So these primary lysosome have inactive enzyme. All the enzyme are inactive in primary lysosome. Inactive enzyme. Just give your attention. So this is the first form lysosome from the Golgi apparatus. Now suppose that this is the food vacuole in amoeba, and in food vacuole food is there. Amoeba capture this food with the help of pseudopodia. Now how this food will digest? Let me tell you how this food will digest. So for digestion of food, these lysosome, primary lysosome and which one? Food vacuole will fuse together. And after fusing, what happened? Lysosome enzymes going to release over food. Are you agree or not? Release over food. So what is the structure form here is called secondary lysosome. And these secondary lysosome now have active enzyme. Now have active enzyme. Got the idea? Now what happened? The food will digest, and after digestion, digested food will absorb. Digest body. So these residual body have which one? Undigested food. Now what happened? These residual body came in contact of plasma membrane and removed the undigested food. So these secondary lysosome, when outside material, food, digest by lysosome, it is forming which one? Heterophagosome. And when lysosome digesting any cell organelle, an example, this is old mitochondria, and this is digested by lysosome, it is forming which one? Autophagosome. So how lysosome have a role in intracellular digestion, I hope you understand. Is it clear in the mind? Is it clear in the mind? Is it done? Understanding occur? Primary lysosome, one physiological state, secondary lysosome, another physiological state, residual body or tertiary lysosome, another physiological state. All 3 have different structure in different physiological state. Na? Understanding the idea? So that is the reason I wrote it is a pleomorphic organelles. Is it clear? Is it done? So now we are going to discuss about which cell organelle? Vacuoles. What we are going to deal? Vacuole. Give your attention. So if we talk about vacuole, we are going to discuss three different type of vacuole. One is large central vacuole, and another is food vacuole, and the next is what? Contractile vacuole. Got the idea? Right. Now I am going to talk about large central vacuole. It is present in plant cell. And these occupy 90% volume of cell. If you want to draw it, you just have a look. Assume it is a plant cell. So in this plant cell, 90% of the volume of the cell is taken by vacuole. This is the vacuole. And it is large central vacuole. So in large central vacuole, two components is there. One is membrane, and the membrane of the vacuole is called tonoplast. So what is tonoplast? It is the membrane of the vacuole, and inside the membrane of the vacuole, the fluid which is present is called tonoplasm. Tonoplasm, or it is also called vacuolar sap. Vacuolar sap, or it is also called cell sap. Got the idea? And the fluid and everything present outside of the vacuole here, this is what basically cytoplasm. Cytoplasm. Now I am writing the composition of vacuolar sap or cell sap. So vacuolar sap, I am writing the composition. Give your attention. Just give your attention. So composition of vacuolar sap. So NCERT wrote it contains water. It contains sap. It contains excretory product. And it contains other unwanted material. Other unwanted material. It contains other unwanted material. So this is the composition of vacuolar sap. Now, one more thing you keep about vacuole in mind is, that is very important, that is vacuolar sap or cell sap is hypertonic over cytoplasm. So who have a more concentration gradient? Vacuolar sap is hypertonic, more solute concentration, and cytoplasm is hypotonic. Cytoplasm is hypotonic. Are you getting the idea or not? Now question arises, how it is possible? How it is possible? Once again, with the help of which protein? Pump protein. So here it is a pump protein. Pump protein means against the gradient. The protein who work against the gradient is pump protein. So it is actively pumping solute. Who is pumping? Actively pumping solute into the vacuole from the cytoplasm. And in such a way, the concentration of vacuolar sap is hypertonic over which one? Cytoplasm. So who is over? Large central vacuole is over. Now, after the large central vacuole, we are going to discuss about food vacuole. As I told it stores food. I have seen in amoeba, In protistans, amoebans, we have seen wherever intracellular digestion is occurring, what is there? Food is stored in a vacuole and it stores food. And contractile, so where it is present? Example, where food vacuole present? Should I write amoeba? It is example of protista. And contractile vacuole, same example I am writing amoeba. So these contractile vacuole perform osmoregulation. What is osmoregulation? To maintaining solute concentration in the cell. So in your body, who do the osmoregulation? Tell me. In your body, who do the osmoregulation? Which human organ do the osmoregulation? Kidney. So who work like kidney in amoeba? Contractile vacuole. Can I say this contractile vacuole is analogous to human kidney? Human kidney. Because it is maintaining the osmolarity in which one? Amoeba or some protistans. Getting the idea or not? So can we say it is maintaining the osmolarity, hence it is analogous. Analogous means functionally same. Analogous means functionally same. So now who is over? Vacuole is over. Now we are going to move about the next cell organelle, mitochondria. Give your attention. Mitochondria. So if we talk about mitochondria, it is also called powerhouse of cell. House of cell. Why it is also called powerhouse of cell? Because associated with energy production, right? Directly ATP production, no problem. ATP is the coin of energy, associated with energy production. Another things I am writing, it is a semi-autonomous cell organelle. It is a what basically? Semi-autonomous cell organelle. Why it is semi-autonomous organelle? Why? Because it have enzyme or protein synthesis machineries. Protein synthesis machinery. It can synthesize some of the required protein. That is, it can synthesize some of the required protein. Not all the protein. If can synthesize all required protein, it become autonomous. But it not synthesize all the required protein, it synthesize some of the required protein, and hence it is which one? Semi-autonomous cell organelle. So machinery is in what present in the machinery? What present in protein synthesis machinery? For protein synthesis, what required? DNA required, RNA required, ribosome required. Got the idea or not? And enzymes required. So these all are present in which one? Mitochondria. Otherwise, if anyone is not there, who will not form? Protein synthesis not occur. Understanding the idea. And of course, you better know, I already told, in mitochondria, ribosome is which type? 70S type. Got the idea? Mitochondria is a 70S type. We already talked now. Is it clear in the mind? Now we are going to talk about more about the mitochondria. So I have given the justification of why it is a semi-autonomous cell organelle. Now I am going to write more. So if I am talking about numbers of mitochondria, it is variable in number. variable. More mitochondria present in physiologically active cell. So the cell who are more active have more mitochondria. The cell who are less active have less mitochondria. Even in your body, each cell don't have same number of mitochondria. Your muscles have more number of mitochondria because it is working. So they need more energy, more ATP production. Getting the things or not? More ATP production, more mitochondria. Are you getting the idea? So it's depend upon which one? Physiological activity of the cell. Is it clear in the mind? So number is variable. Then after that I am going to talk about size of mitochondria. If we talk about size of mitochondria, diameter, average diameter is 0.5 micrometer, and if we talk about length is 1 to 4.1 micrometer. These are the data mentioned in your NCERT, and this is which one? Average diameter. Average diameter. Got the idea? Clear in the mind? So size we have seen about. Now we are going to talk about shape of mitochondria. Shape of mitochondria. So if we talk about shape, it may be sausage shape or cylindrical in shape. Got the idea? Now we are going to talk about the structure of mitochondria. So it is a double membrane bounded cell organelle. Cell organelles. Now I am going to say you about the structure of mitochondria. So I am drawing the structure of mitochondria. Give your attention. So here suppose that this is a mitochondria. So I have drawn here a which kind of mitochondria? Cylindrical mitochondria. Which kind of mitochondria I have drawn here? I have drawn here a cylindrical mitochondria. What is the idea? Suppose that it is a cylindrical mitochondria. So it have two membrane. One is outer membrane and another one is which one? Inner membrane. Inner membrane of mitochondria is highly enfolded. It is highly enfolded. And these highly enfolded inner membrane is called cristae. Another name is what? Cristae. So I am just writing here. It is which membrane? Outer membrane. Outer membrane of mitochondria. And this is inner membrane. Inner membrane. And this inner membrane is also called cristae. So what did cristae do? Increase the surface area for cellular respiration. Increase the surface area for cellular respiration. Any enfolded area, increasing the surface area, cellular respiration. And now if you talk about membrane, what is important to know? Both membrane contain different enzyme. Both membrane contain different enzyme. Means their function are different. That is, function of both membrane are different. Function is different. Got the idea? Inner membrane is less permeable. Is less permeable than outer membrane. Anybody can answer it? Why inner membrane is less permeable than outer membrane? I am expecting you will answer it. Please answer. Pin your answer. Why inner membrane is more permeable than outer membrane in mitochondria? Please pin your answer. I am giving you help. Hint. Connect with transport in plant. Something written in porine about porine. In transport in plant, something written about porine. Yes, na? Where porins is present? Inner membrane or outer membrane? What NCERT wrote? Porins present in outer membrane, na? So due to presence of porins, outer membrane is more permeable. And these porins are not present in which one? Inner membrane, hence it is less permeable. Answer came? Answer came. Now I am going to move ahead about in between outer membrane and inner membrane there is a space is there. It is called perimitochondrial space. Perimitochondrial space. And toward inner side also there is a space is there, and in that who is there? Fluid filled matrix is there. Who is there? Fluid filled matrix is there. Matrix is there. Inside the matrix who is there? Double stranded circular DNA is there. Another structure present inside the matrix is what? 7 मैं भी क्लास के बाद बताता हूँ। बिना माइक के भी तो पढ़ा सकते हैं जी। अरे बिना माइक के भी तो पढ़ा सकते हैं।
या
हाँ, बिना माइक के बताइए, हो जाएगा?
हाँ, नहीं आगे वॉइस तो अच्छा नहीं,
जो भी आता ना,
जो भी आता है।
हाँ। So look, give your attention. Look here what I told. Here in diagram I have drawn a cylindrical mitochondria. Which shape? Cylindrical shape. So it have two membrane. One is outer membrane, another is inner membrane. Inner membrane is highly enfolded, and this enfolded inner membrane is called cristae. Now this enfolded inner membrane increase the surface area for cellular respiration and enzymatic action. Yeah. Now, it's came audible? Okay. Haji. So what I am saying, outer membrane, inner membrane. Between outer membrane and inner membrane, there is a space called perimitochondrial space. Inside the inner membrane, this is the space where fluid-filled matrix is there. In this matrix, DNA is present. In matrix, 70S ribosome is present. In matrix, some few RNA is present. And in matrix, enzymes are present. These are the enzymes for protein synthesis as well as link reaction and Krebs cycle. And if you look at the inner membrane, you are finding pinhead knob-like a structure present at every 100 angstrom, and this is called elementary particle or F node and F1 particle or oxysome. Getting the idea or not? So if you look the detailed structure of this, it is looking like this. It is inner membrane, F naught, F1, F naught particle. Here it is F naught particle. This is F1 particle. So these are present in the inner membrane. About that we will deal in much detail in which chapter? Cellular respiration. If we talk about the function of mitochondria, so aerobic respiration. If mitochondria is not present in a eukaryotic cell, aerobic respiration not occur. But in mitochondria, if mitochondria not present in a prokaryotic cell, beside of that, aerobic respiration may occur because mitochondria like work done by mesosome. Got the idea? Now I am telling function of mitochondria, aerobic respiration. Link reaction and Krebs cycle of respiration occur in matrix, electron transport system and oxidative phosphorylation occur in which one? Inner membrane of mitochondria. Second function is what? Cytoplasmic inheritance. It is also called maternal inheritance. Your mitochondria in your body, either boys or girl, doesn't matter. Always contributed from your mama, not from daddy. Because who contribute the cytoplasm in zygote? Mother. So mitochondria present in your body exclusively came from your mother. Got the idea? So it is responsible for cytoplasmic inheritance, and hence this is also called maternal inheritance. Got the idea? So this is about which cell organelle? Mitochondria. Right? Okay, ji? Done? So I think this is the second class of the week or third class of the week. Probably second class, na? It is second class of the week or third? Someone writing second, someone writing third. I also feel like it is second of this week. Okay, whatever is it. Third. Third, okay. So 2 more classes there. Probably it's required to take, if it is needed, I will take extra class for your batch. We will inform you. Okay, ji? Right? Bye-bye. Have a good day to all of you. Bye. So this is the QR code. Thank you very much for your patience, for listening. Bye-bye.